So for the forensic analysis literature review, I chose an article that focused on essentially seeing if they can devise a method for predicting the age of individuals with small samples of DNA, specifically those less than 10 micrograms. And so the title of this article was Age Prediction by Methylation Analysis of Small Amounts of DNA Using Locked Nucleic Acids. This article was received July 28th of this year, revised September 2nd, and was ultimately accepted September 13th, 2022. So this is very recent literature. So for the background of age prediction by methylation analysis, ultimately what this is it is looking at DNA to determine how old an individual is. And we're able to do this because DNA of older individuals tends to be less methylated than those who are younger. And so what we would typically see is that older individuals would have less methylation, which is what we're looking for later on in the research. So typically DNA is methylated at sites where a guanine follows a cytosine with an intervening phosphate group. And these sites are referred to as CPG sites, and there are about 20 million of these in our DNA genome. And so there's a lot of locations where this occurs. So how exactly can we use methylation of DNA to then predict age? Here, what they did is they focused on certain genes that have demonstrated a correlation with age. And so of the options that current literature has provided, they chose to focus on the KLF14 gene, the EDARADD gene, and the SST gene. So previous methods for these required a minimal DNA sample of 10 to 20 nanograms. And so this is a really small sample of DNA, but sometimes at crime scenes, there's even smaller samples of DNA. And so what they were wanting to do is fill this gap of, if we found 5 nanograms of DNA, how would we be able to analyze this? Can we still determine someone's age? And so they looked at using something called locked nucleic acids to do that. So now the question arises, how do we actually methylate DNA? And so there's multiple different methods for DNA methylation. There is whole genome methylation profiling, searching for differentially methylated regions, bisulfite conversion, which is what was used in this research article, and a digestion-based assay after a PCR. So before we kind of go into bisulfite conversion, it's important to know what DNA methylation is. DNA methylation is a chemical modification of DNA where methyl groups are added to the DNA molecule. And this happens naturally in the human body. And as mentioned previously, elderly people are going to have lower rates of methylation naturally in their body. That's a sign of age. So when DNA is methylated, it changes the activity of genes or regulatory elements without actually changing the sequence. However, with bisulfite conversion, you are changing certain nucleotides from one to the other. So it's a little bit different with bisulfite conversion versus naturally in your body. So bisulfite sequencing is considered to be the gold standard when it comes to DNA methylation in the lab. And so essentially what bisulfite conversion does is it converts unmethylated cytosines into uracil. So as individuals age, methylation decreases. So we would expect to see more unmethylated cytosines, which then get converted into uracil, which then gets converted into thymine. So as someone's age were to increase, if we were doing bisulfite conversion of their DNA, we would expect to see more thymine than cytosine, because the thymine is what results for an initially unmethylated cytosine, whereas methylated cytosines are then converted into guanine. Additionally, bisulfite conversion can reduce genome complexity, and this is due to if every single cytosine is unmethylated and it's then ultimately converted into uracil, you've lost cytosine from that sample. However, in the samples for this research study, not every base was unmethylated, not every cytosine was unmethylated, and so therefore it gave us a percentage of unmethylated cytosines that eventually became thymine and methylated cytosines that were converted into guanines. Earlier, it was mentioned about locked nucleic acid primers. These are really important, and this is what the researchers focused on, and I'll go into why that is. So locked nucleic acids are man-made RNA nucleotides that are cross-linked. And so what that means is there is an additional bond connecting the 2' oxygen to the 4' carbon. And when this happens, it actually increases the stability of the nucleotide. So they are very thermally stable due to their reduced conformational flexibility, their inability to move as much. And so due to them being thermally stable, this actually improves amplification efficiency. And then additionally, if we incorporate LNAs into DNA, this helps us raise the melting temperature value and again, increases stability, which is really important when gathering our data. And so stable binding to the DNA and PCR increases the height of the peak for genes and improves the accuracy of methylation analysis. So ultimately, by using these LNAs, the researchers were trying to figure out, does this make our method more statistically significant? Are we receiving accurate results? And that will be discussed later. So for this research, they had a 5-step method, with the first step being the bisulfate conversion. So in order to do this, you have to first denature DNA. And so this is just essentially making DNA single-stranded, and then it needs to be treated with sodium bisulfite. This sodium bisulfite converts those unmethylated cytosines into uracils and leaves methylated cytosines unchanged. Then that is amplified by treating the DNA with PCR, and uracils are further converted to thymines. So at this point, all nucleotides that were unmethylated cytosines originally have become thymines, while those that were methylated cytosines still remain cytosines. So this is really important for these researchers because as older individuals have less methylation, it's expected there would be higher amounts of thymine in their samples and lower amounts of cytosine, as the sodium bisulfate does not alter the methylated cytosines. The second step of their method was designing primers. They initially targeted 4 age-associated genes: the PDE4C gene, the EDA-RADD gene, the SST gene, which is somatostatin, and the KLF14 gene, but as they began their research, found that the sensitivity of the PDE4C gene was much lower, and so that one was eliminated, as mentioned earlier. Both normal DNA primers and DNA primers with locked nucleic acids were designed, and the normal DNA primers were used as controls to determine if there was a significant difference between normal DNA primers and those with the LNAs or locked nucleic acids. And the primers that had the LNAs contained 3 of those in place of 3 normal DNA nucleotides. The third step was amplification of the DNA, and they used multiplex PCR to do this. The fourth step was a single base primer extension reaction, which is an SBE reaction. This was an effective and sensitive tool that helped them type their loci throughout an organism's genome in a single reaction. And then lastly was the detection and data analysis component, and they used relative fluorescent units, which are RFUs, to assess the intensity of signals from unconverted methylated cytosine and converted unmethylated cytosine. And so those 2 components allowed them to then correlate that sample with a given age based on how many unconverted methylated cytosines were present and what percentage of converted unmethylated cytosine was present. So for the results, they found that the coefficient of variation, or CV, increases as DNA sample size decreases. This makes sense because as you have less DNA available, you're going to see more variation because there's just less sample to work with. However, they did find that locked nucleic acids reduced this variation. And so this already shows that by using locked nucleic acids for their primers and only using 3 of those in said primer did reduce the amount of variation seen, which is significant for further research. Additionally, methylation levels were positively associated with age in the SST and KLF14 genes and negatively associated with the EDA-RADD gene. So this was kind of just a check to make sure that these genes did in fact associate with age, and all 3 were correlated, 2 being positively correlated and 1 being negatively correlated. Additional results were that the ability of these genes to predict age were similar in both DNA and LNA primers, but LNA primers improved age prediction accuracy for all amounts of bisulfite converted DNA. And this is really important because sometimes at a crime scene or when collecting evidence, there's not a lot of DNA available. And so what this is telling us is that, if we have less than 10 nanograms of DNA, for example, if we have 2 nanograms of DNA, there will be more variation when it comes to predicting age, but the use of LNAs could allow for more specific and targeted analysis. So it would have a wider age range with the smaller samples of DNA, but it would still be more accurate than our current DNA analysis without using those locked nucleic acids. And another important result was that signal intensities increased for the samples using the LNAs versus the samples that used the normal DNA primers. So as for legal considerations, the ultimate question everyone has is this method admissible in court? Well, unfortunately, the answer is not yet. This was a research article that was published on September 13th, 2022, so this is extremely recent. And so further research needs to be developed and investigated before this can be introduced into a courtroom. However, this method could potentially be used to determine the age range of a potential perpetrator or even a potential victim. For example, if a small blood stain were found at a crime scene, locked nucleic acid primers would allow for an age range of that individual to be determined. And so as this technology is further developed, hopefully we could narrow that age range down to be more specific, which could then help us find a potential victim or locate an age range of the perpetrator. However, as it was stated earlier, this technology is so new, as it was published September 13th, 2022, that further research does need to be done. It usually takes larger samples of DNA to complete an accurate age prediction analysis, and those larger samples are usually greater than 10 nanograms of DNA. But this could change the way that works. This could revolutionize DNA collection and the fact that we would need tiny amounts of DNA to be able to predict that a donor's age. If this were presented to a jury, there would need to be extensive scientific explanations, as this is not only a new technology, but this is a very in-depth technology that would require a lot of explanation and visuals to provide the jury so that they accurately understand the technology and what it can tell us. It's important as well to mention that there is an age range and there's also a coefficient of variation. So there is no set age. Therefore, we can't say this person is 82 years old. It would need to be an age range. Ultimately, this is such a new technology that it's not admissible in court just yet. There needs to be additional research to substantiate and build on the research presented in this article. So some loopholes and limitations is, again, this is a very newly developed technology. If this were introduced into a courtroom, it would be not only very confusing because the general public does not understand the terminology and just methodology, but also it's so new that even experts in the field may not know about this yet. So because of this, there needs to be more research and larger sample size. This study was conducted in Japan and it used 58 individuals, which is a relatively small sample size. And so we would need to conduct more extensive research with many different populations of many different races, many different ages, just to determine if this applies across an entire spectrum of race and age and diversity, as it was only conducted in one specific race, in one specific area, and a small sample size of 58. However, there's still a mean absolute deviation of 4.94 years for a sample size of 10 nanograms. So even if we were to have 10 nanograms of DNA, that would still have to be an age range of around 5 years, which is a pretty large age range considering. There could be conditions that could alter the methylation of DNA. So there are certain genetic disorders that affect methylation of DNA. And sometimes if someone were to have one of these, the results would not be as expected. So, for example, someone older would be expected to have higher levels of unmethylated cytosines, but if they were to have a certain genetic disorder that caused that to not happen, that would throw off what our expected results would be. And then lastly, LNAs, or locked nucleic acids, are not effective for methylation analysis of all genes. So as mentioned on one of the first slides, there are certain genes that have been associated with age, and there are certain genes that have been associated with being able to be methylated and then determine if that methylation correlates with age. So unfortunately, LNAs cannot be used on every single gene. There is a small subset of genes right now, given current research, that they can be used on. So hopefully with further research, there could be more ability to use LNAs on other genes than those mentioned previously. Lastly, the implications and future use of this technology is incredibly important. We could potentially use extremely small samples of DNA, again, those less than 10 nanograms, to determine the age of a donor. And not only could we do this with a really small sample, we can also do this with many sample types. And so the application of LNAs can be used on saliva, blood, teeth, bones, and even bodily fluids. So regardless of what our sample is, we could use LNAs to determine the age of that donor, which is huge because that could then provide forensics and detectives an avenue whether they're looking for the age of their perpetrator or the age of their victim.